Apo is a poor molecule all by itself. You add a binding partner and things get complex. And if you add a partner that something needs, well, now you make it holo. So apo, complex, and holo are three words or word parts that you might see in biochemistry. Most commonly with apo and complex, we're dealing with structural biology. So basically structural biology is this field of biology where you try to use methods like crystallography and cryo-EM and NMR to get a look at molecules at their atomic level, at their 3D shape at the atomic level. And often we don't just want to look at one protein. We want to look at how proteins are interacting with another protein, or how proteins are binding to a drug. Look at that target site and see, okay, well, the drug binds like this. Can we make the drug a little better so maybe it can bind better, it can have better specificity, it can overcome resistance? To do all these things, if we know the shape of that binding site and we know how they're interacting, we can better design these molecules. Then you might just want to see how proteins are interacting with one another, how they're interacting with nucleic acids, so DNA and RNA, how they're interacting with lipids. You might want to see how an enzyme is interacting with a substrate, so the thing that it modifies. There's lots and lots of reasons that you wouldn't want to just look at a molecule by itself, so its apo form, but you would actually want to look at a complex, so that protein or whatever molecule you're looking at bound to another binding partner. And so sometimes we call these binding partners ligands, and there's a whole bunch of ligands that there could be. And there could also be, typically when we have like two proteins bound to another, we don't really call them ligands. We typically use ligands for more small molecule type things, like if a receptor binds to something, that something would be a ligand. But ligand just means a binding partner. And we're talking about an enzyme, so a protein, typically a protein, sometimes RNA, sometimes both, that modifies something else. It helps catalyze a reaction. Then the substrate is like the ligand that it binds, but it actually changes that thing. So that's a special case of ligand. But for all these things we can be talking about, when we have multiple molecules bound together, we can call this a complex. And it can be really useful to get a look at these. But it can also actually be complex and kind of pun intended, but really it can be hard. And so there are different strategies that people use in order to try to figure out the structure of these complexes. And so say you want to solve the structure, so basically figure out the 3D shape using crystallography or cryo-EM or something like this. In this case, let's talk about crystallography. Say you want to solve the structure of a protein bound to a drug. Well, there are several different strategies that you can use. So crystallization, just a quick recap, I have much more on this in other posts, but basically you have these molecules and you get them to all freeze. And so come out of solution, so they're all floating around in these water coats, then you get them to freeze in place in this lattice. So basically you have these repeating units of these protein or whatever it is over space, and then you shoot x-rays at them, and those x-rays, they bounce off the molecules. It's not quite that simple, but you can think of it kind of like that. And then you end up with these like diffracted waves. So the x-rays, they interact with one another, and then it ends up, you get this pattern of spots called the diffraction pattern, and you work backwards from that. So for all of that to happen, you have to have that crystal, and getting that molecules to crystallize can be really, really hard. And what you can do though is that if you want the structure of a protein with something bound to it, you have a couple options. One is co-crystallization. Here you actually mix the protein with the drug or with the other molecule you want it to bind to, and then you try to get them to crystallize together. And so sometimes this works, sometimes it doesn't. And if you have, say, something that's really expensive, it might be hard to try to get it all to co-crystallize because you're trying a bunch of different conditions and you might be using up a bunch of your really expensive stuff. So an alternative is soaking. Here you get the crystal to form, so you already know, okay, well, there's a crystal here, so it's worth adding my drug. And then the drug can kind of sneak into the crystal, because although it's like this solid, it's not a crystal like your diamonds. Instead, it's this kind of, it's the soft crystals, so sometimes if you touch them, they kind of like crush, which is not happy, but except that it tells you that it's like a protein crystal and not salt. But basically, inside of these crystals is actually these channels of water. So these protein crystals are really like 50% or so water. And so you have these water channels flowing throughout. And those little drugs can flow in there and bind as well. And so sometimes this works, sometimes it doesn't. Sometimes a protein is actually more stable when it's bound to a substrate, when it's bound to a binding, or that'd be an enzyme, but when it's bound to a ligand, when it's bound to something, sometimes that stabilizes the protein. If the protein's moving around too much, that can make it hard to crystallize or to get a cryo-EM structure or something like this. But binding to a partner kind of locks it more in place. And so sometimes you can actually have better luck getting a complex to form than the apo form. And so it can actually be harder to get the apo form to crystallize. So when I was in grad school, I was working on this protein called argonaut. And basically, you couldn't get its apo form, so its unbound form to bind. So it bound RNA, these microRNAs. You couldn't get that apo form to crystallize, but you could get the RNA, the microRNA bound form to crystallize. So sometimes things are easier to crystallize in the presence of some sort of binding partner, but of course the binding partner is going to depend as well. The other option is that if the thing is like present in the cells, so if you want to purify some like native complex, so you want to instead of drugs, let's think about, okay, there's some small molecule in the cell, some metabolite, some like intermediate of some molecular making or breaking, and it's bound. Or you can think about something like ATP or something like glutamate or something like this bound to a molecule. Well, if it has high enough affinity and there's high enough concentrations of it inside of the cell, you might be able to actually purify that complex out of the cells, and it's like this native complex. So basically just take it as it is. But again, this isn't going to work in all cases, and it only works if you have a really high affinity and you have plenty of the thing that most of what you pull down is going to actually be bound. So that was if we were binding to a small molecule. Now let's talk about macromolecule scale. Let's talk about proteins. When we talk about macromolecules, we're typically talking about like proteins, we're talking about we're talking about RNA, DNA, lipids, all this stuff. Often we're talking about, we want to see a structure of proteins bound to one another, because proteins, they typically don't work by themselves. Sometimes they do, but often they work in concert with other proteins, even if those other proteins are kind of just holding it in place, or holding it to another protein that it needs. So some sort of like scaffolding protein helping hold these things together. If you want to get a complex of multiple proteins bound together, there are different strategies that you can take for this too. So one of them is similarly to how we could purify the native complex out of the cells for the small molecules, if those were present in the cells. We can also purify native complexes out of cells of these proteins bound to one another. But proteins typically don't just hang out with one other protein, they might hang out with other proteins, and they often hang out with one protein sometimes and other proteins sometimes. So it can be hard to purify out just a protein with a single binding partner that you want to look at without getting all these protein with this other binding partner and that sort of thing. So often structural biologists turn to doing like exogenous expression. So instead of just taking the native complex, we actually make a modified version of the protein where we do something like we stick an affinity tag on the end, something like a his tag or a strep tag. Basically, this is just a little extra sequence at the end that's going to allow it to bind specifically to this little column of beads, and then we can flow things through and those will get stuck. We can wash everything else off, and then we can wash our stuff off or elute it, and then we get our protein that had the tag without everything else around. And if that protein that had the tag was bound to something else, well, then you also get that something else. And so if you do this, what you can actually do is you can actually like co-express proteins. So you can purify the proteins, both of them in the cell together. Sometimes you might tag one of them if it's like a complex where you typically only see these two hanging out and you don't get a bunch of that other stuff. Or if you want to make sure that you're only getting the complex with just those two things, or at least containing those two things, you can tag them both with different tags and then use one tag and then use the other tag and things like this in order to get that complex with them both together. So in that case, we're co-expressing them. We're expressing them in the same cells. And when we say express, basically we're just getting the cells to make them. And so we can get the cells to make them. Another benefit, even if you could purify it out native, even if it's like just the complex is normally hanging out together, so you don't have to worry about those other like side complex type things, still you might not have very high concentrations of those molecules inside of a cell. When you do an exogenous expression, when you make the cells make something for you by putting in the instructions for making it, you can get the cells to overexpress it or make more of it than it usually would. And this is going to allow you to have higher concentrations as well as potentially have that tag to make it easier to purify. And so those would be cases of co-expression. Now, sometimes with co-expression, you could get uneven levels of the proteins being made. So sometimes the cells will be really happy making one of the proteins, not so happy making the other protein. And so in these cases, what people might do is they might actually make a fusion protein. This is also can happen if your proteins don't stay together very well. The complex isn't that strong. What you can do in this case is you can take the genes for those proteins. Well, more specifically, you're taking the cDNA, so kind of like the DNA version of the messenger RNA, so it's the edited version, and you're kind of putting them back to back. And then you put a linker in between of them so that they have this flexible spacer. And now these proteins are made as one long chain and they each fold up. And as long as that linker is long enough and it's not like hidden inside the protein, well, now these molecules are able to form that complex but actually be physically stuck together. So this fusion strategy also sometimes works. And then finally, what you could do is you could just express and purify those proteins individually. This works sometimes. This is a common way that's actually used because you can modify the proportions of the different ones. So if one of them doesn't express that well, but one of them expresses really well, you can basically put that one that doesn't express that well with like a ton of the one that expressed really well, and then purify out the complex using something like size exclusion chromatography, where you basically can then isolate the bigger thing that corresponds to your complex being formed. And by using these really high concentrations of the thing you have a lot of, well, now you're able to kind of maximize the chances that that thing you have a little of, you'll get most of it being in the complex. So the strategies for the multi-protein complexes are like just purify it from its native form, go express them and purify them individually and mix them together, or express them as a fusion protein. Now finally, before I go, I want to make this post-holo and talk about the term holo. So apo, that was like without or lacking, and holo, this is whole. If you typically see the word holo in context of enzymes. So enzymes, remember, are these reaction helpers or speeder-uppers. And so instead of just binding to something, they bind to something and they change that something. And we call that thing that they're binding and changing the substrate. And they might bind other things as well, though. They might need cofactors, which are kind of helper molecules that bind to them. Things like vitamins sometimes serve as cofactors. There's a lot of like vitamin B and that sort of thing. When an enzyme needs one of these cofactors, if it doesn't have that cofactor, it's not active. And if it doesn't have this cofactor, well, now it's lacking that cofactor, so it's the apoenzyme. If you add that cofactor, you add the helper molecule, now you made it whole, you've got the holoenzyme. And so holo is often used in the context of these enzymes. And anytime you have multiple molecules bound together, now you have a complex. And sometimes getting these complex structures of these complexes can be complex, but sometimes it can actually be easier than getting the apo or the solo form. So I hope that helped you understand these terms as well as some of the strategies that scientists use in order to study apo and complexes, things.